Okay, so we will get started. Hi, everyone. Welcome so much to today's Insect Welfare Research Society research seminar. We are delighted to be welcoming a 2024 Graduate Research Award winner in welfare. Jesi Gibbs is joining us to share about her work on pain in shrimp. And also, Jesi Gibbs has just earned her master's degree, I believe, from the College of Charleston. So congratulations as well on defending your dissertation. Just a few notes for our audience members. You can ask questions at any time. You'll just pop down to the bottom of your screen where you'll see a little Q&A button. You can click that and it'll post a question in our Q&A channel. I will have access to those questions and I will go through and ask as many as we have time for at the end of the talk today from Jesi. So feel free to answer or ask your questions at any time. And with that, I will turn things over to you, Jesi, to get us started.
Thank you so much, Megan. I think you all can hear me. Yes, great. Thank you all so much for being here. Like Megan said, today I'm going to present to you my master's thesis research that I performed at the College of Charleston in South Carolina, which was supported in part by a grant that I received from the IWRS about a year ago. So my research concerns pain perception in shrimp. Now, a common argument used to dismiss the idea of pain in animals is that behaviors that look like pain are only reflex. The signal isn't centrally processed and the animal doesn't actually feel anything. But I'm going to show you here today that when a shrimp is subjected to a noxious stimulus, it also results in this thing we call nociceptive integration, which is evidence that they're capable of feeling pain. So as many of you may know, pain research is crucial for animal welfare because globally, the language of welfare legislation is often based on sentience. If you're interested in the welfare legislation, this topic, Browning and Birch have a great review of this issue in their 2022 paper. Sentience can be defined as the capacity to feel hungry, content, joyful, and most importantly for animal welfare, to feel pain or suffering. These welfare laws typically require humane treatment of sentient animals. So humane husbandry and slaughter methods, anesthesia for invasive procedures, and ethical pre-approval for research proposals. And science actually plays a really large role in deciding which animals we consider sentient. So based on a wealth of evidence, most places globally have at least some protections for vertebrate animals used in research or for food. But only a handful of jurisdictions protect invertebrate animals. Based on some recent evidence, several jurisdictions now consider octopus welfare, and a few have come around to the inclusion of decapod crustaceans. Oops. Notably, the UK just added these animals to their Animal Welfare Act a few years ago, based on a report by Jonathan Birch and his team, who reviewed relevant literature, determined there to be compelling evidence for sentience in these animals, and recommended the protection of all decapod crustaceans under the precautionary principle. However, most of the research has been done on the infraorder Reptantia. This group includes crabs, lobsters, crayfish, and hermit crabs. But decapods are a really diverse group, and some have suggested that maybe not all decapods are sentient. In particular, there's this big evidence gap for shrimp. In the UK review that I previously mentioned, and the follow-up article published by Andrew Crump and colleagues, authors list these 8 criteria for sentience that are likely very familiar to some of you already. In brief, the more of these criteria that are fulfilled with higher confidence ratings due to more research in the area, the more likely the animal is sentient and deserves welfare consideration. So you can see that shrimp overall have a lot of low confidence ratings, which the authors argue is mainly due to a lack of research in this area. Pinnate shrimp have been researched the least in terms of pain or sentience, but for the Caridian shrimp, really only a handful of studies have been done to contribute to the ratings that you see here. This evidence gap really matters because we love shrimp. Actually, to find the pictures of the shrimp that I used on this page, I had to search alive shrimp, or all you get are photos like these. So we eat a lot of shrimp. This figure here from Rethink Priorities shows average estimates of shrimp and other resource animals killed each year globally. So more shrimp are systematically killed each year than any other animal, and more are alive at any time on farms than any other animal. Now, I will say that with the scaling of the insect farming industry, insects are projected to surpass shrimp in the near future on these metrics. But yes, we kill an estimated 27 trillion individuals, shrimp individuals each year. Additionally, decapod crustaceans, including shrimp, are increasingly used for biomedical and ecotoxicological research. So if these animals are able to feel pain, there may be a tremendous amount of suffering occurring on the individual level that deserves our attention. Now, what is pain? How do we recognize it as scientists, and what could it possibly be good for? Well, it starts with nociception, which is a sensory capacity to detect damaging or harmful stimuli. The function of nociception is to evoke a reflex response to get the organism away from the harmful stimulus as quickly as possible. The reflex doesn't require the brain to make a decision, so the response can be rapid. You can think of this as when you touch a hot stove and your hand jerks away from it before you really process consciously what happened. In some animals, nociception also results in a subjective experience or a feeling, which we call pain. So moments after your hand jerks away from that hot stove, you feel it, and you'll likely continue to feel it. And that's because that nociceptive signal has moved from your hand to your brain, where it's combined with other sensory information in a process known as nociceptive integration. And this integration creates that felt experience. Pain, in contrast to nociception, is highly adaptive in the long term. So the negative feeling of pain alters motivation to promote survival while injured and reinforces learning so that you don't repeat the same harmful behavior twice, hopefully. So in response to the pain in your burnt finger, you will likely ice and bandage your wound, avoid using it until it heals, and hopefully you will have learned not to touch hot stoves again. So the difference between nociception and pain is that pain involves the brain to produce these longer lasting fitness benefits. Thus, evidence of nociceptive integration suggests a capacity for pain. So I wanted to answer the question, do shrimp display nociceptive integration, making it possible that they feel pain? So I used two approaches in this project, examining both the shrimp's behavior and their brains. Through observation of shrimp behavior, I wanted to learn, can shrimp learn from nociception? And do shrimp display non-reflexive behavior in response to nociception? Again, getting at some of those adaptive functions that we would expect to see from pain, which are reliant on that nociceptive signal making its way to the brain. On the neuro side, I wanted to back up that behavior study by asking, does a pain-associated biomarker increase in the shrimp brain in response to nociception? And is the effect localized to areas of the brain involved with learning, memory, and sensory integration? Or those areas of the brain that might be involved in producing the adaptive effects of pain, as well as the felt experience of pain. For my experiments, I used the study subject Alpheus angulosus. They are a snapping shrimp, aka a pistol shrimp. This is a Caridian shrimp. They are local to the southeast US coast. They have asymmetrical claws. So the big one makes a snapping sound, hence snapping shrimp, which shoots a jet of water, hence pistol shrimp. And the smaller claw is for grasping and burrow building. Alphiids have really large and complex brain structures, so they're a good choice for a learning study. And finally, they are really easy to keep in the lab and really compliant research subject. One of my advisors had used them extensively in behavior studies previously to this. And unfortunately, of course, to study pain, I needed a stimulus that would inflict pain. So I chose to use noxious heat. I used a wood-burning iron plugged into a variable transformer so that I can control the temperature. Heat has been commonly used in pain studies with mammals. And Puri and Falks showed in 2015 that heat is nociceptive to crayfish. So I thought it was a good choice for this shrimp study. Across experiments, I utilized two treatment conditions. The hot condition, which was the iron set to about 65 degrees Celsius. And the control condition was the same iron turned off at room temperature. I delivered my experimental treatments as touch bouts, in which I touched the animal between the propulus and dactyl on the small claw 3 times. I used touch bouts for all experiments. So anytime we're comparing hot and controlled groups or conditions, we are comparing the effects of hot versus controlled touch bouts. In order to validate any claims about pain or nociceptive integration, I first need to show evidence that I caused nociception. So with the hot iron. So during my behavior and my neuro experiments, I performed the experimental treatments, the touch bouts, and I recorded them on video. And from these videos, I used both shrimp behavioral reactions, their reflex reactions, as well as human observer reactions to their behavior to gauge whether the hot iron resulted in nociception. We're going to kind of go through this section really quickly to save time, but if you want to ask more about it later, I'm happy to answer questions. So I first measured instances of reflex responses to the touch bouts. I used 24 subjects for these experiments here, and I used repeated measures for these trials. And I counted tail flips and dropped claws to compare hot and control conditions. And it turns out that the hot iron results in more nociceptive behavior than the room temperature iron. So subjects perform significantly more tail flips when touched by the hot iron. There were also 3 total dropped claws during all experiments. They were all in response to a touch from a hot iron, and all dropped the claw that was touched by the iron. Additionally, in order to measure behavior that was not easily countable, like the tail flips and dropped claws, I devised an experiment using human observer responses to videos of shrimp reactions to the iron. So for this, I used a separate set of videos from my neuro study. There were 12 shrimp in these trials, and each received 7 touch bouts from either a hot or a control iron. I had 21 volunteers, unbiased lay people, watch randomized clips of shrimp behavior reactions to the touch bouts, and they answered 2 prompts for each clip. The first was the iron hot or off? And rate the degree of disturbance caused by the iron on a scale of 1 to 5. 1 being not at all disturbed by the iron, and 5 being extremely disturbed by the iron. And here are the results from those responses. So this graph shows the proportion of hot or off responses from observers for clips of animals in each treatment group. So when the iron delivering the touch bout was hot, observers were significantly more likely to judge the iron as hot based on the shrimp's behavior. The second graph shows the number of responses for each disturbance rating from 1 to 5 for hot and control clips. So observers rated subjects as significantly more disturbed by the hot iron than the room temperature iron. So shrimp behavior, as well as observer reactions to their behavior, suggests that the iron is indeed nociceptive to this species. Cool. So we know that we cause nociception, and now we are ready to look for evidence of nociceptive integration. We're going to start here by looking at behavior indicative of pain. So to reiterate, one of the most important adaptive functions of pain is to learn to avoid something that has previously hurt you. A classic behavioral paradigm for avoidance learning is conditioned place avoidance. This paradigm has been used to study emotional pain and chronic pain in mammals. And Robin Crook recently used conditioned place avoidance along with conditioned place preference as evidence for pain in an octopus. So I wanted to see, is it the same for shrimp? Conditioned place avoidance involves associating a place with a noxious stimulus to detect whether avoidance learning occurs. So here's essentially how it works. My laser pointer. We have 2 distinctly different choice zones. So here we have dark and light zones, as well as a middle acclimation zone or starting arena. Shrimp, like many other prey animals, prefer the dark because it provides shelter and safety from predators. So if you remove these acclimation barriers, the shrimp will choose to spend the majority of their time in the dark zone. In this animal, I did some preliminary trials with scan sampling, and the shrimp spent about 90% of their time in the dark zone with free choice. If, however, I were to touch the shrimp with a hot poker in the dark zone, they might learn to associate the dark zone with that harmful stimulus and give up that preferred safe space and spend more time in the light side to avoid future harm. This change in preference would suggest learned avoidance, which in this case can only occur with the central integration of that nociceptive information. So I predicted that shrimp touched by a hot iron in the dark would learn to avoid it, spending more time in the light and less time in the dark than shrimp touched by a room temperature iron. In my experiments, I used red film to create the dark environment. So shrimp only see in the blue and green, so the red film allowed me to see where they were in the chamber in my videos, but they would have seen that side as dark. So for a single trial in my experiment, here is kind of how it went. I started the shrimp out in the acclimation zone for 5 minutes. I then removed the barrier to the dark side, kind of giving them a simulated choice to enter the dark side, where I performed my touch bout with either the hot or the room temperature iron. I then gave them 5 minutes to create an association between that touch and the dark side of the chamber, at which point I reset them in the middle for 1 minute to keep it consistent between subjects. and then gave them free choice between sides of the chamber for 10 minutes. We did 3 trials in a row for each experiment. So as soon as the individual was finished with its 10-minute choice, I reset it back into the acclimation for 5 minutes, and we did this 2 more times. In order to determine whether they learned to avoid the dark during the choice test, I measured time in each zone, time in the light side, time in the dark side. These are not flip sides of the same variable because we have this middle acclimation area here, which we considered a non-choice zone. So only when they had passed their head and their claws into either the dark or the light zone were those variables counted. I also measured locomotion as an additional measure of non-reflexive behavior. I measured this by measuring time paused, which I defined as any time that they spent not moving for more than 3 seconds. To account for individual variability in behavior, each subject underwent both the hot treatment and the control treatment. So we did 2 rounds of experimentation about 1 month apart to allow the shrimp to molt and for any possible injuries to heal for animals that experienced the hot treatment in the first round. Okay, so let's look at some results from these experiments. So starting with learned avoidance. My first prediction was that subjects touched by the hot iron would spend more time in the light side of the chamber. And as you can see here, there was a significant treatment effect consistent with my prediction. Hot subjects spent more time in the non-preferred light zone than the control subjects. Similarly, as predicted, hot subjects spent less time in the dark than controls. However, for this variable, there was a significant interaction between treatment and the round of experiment. So the effect here is dependent on the round of experimentation. So I analyzed round 1 and round 2 separately. So as you can see here, in round 1, we see no difference between the control and the hot treatment groups. However, in round 2, we see a significant difference emerge between the treatment groups, where the hot group spent significantly less time in the dark zone than the control group predicted. This difference between rounds is kind of odd, but it might be due to a reduction in novelty of the trial. The shrimp were familiar with the paradigm in the second round, perhaps allowing for more natural behavior to occur. Or, of course, it could be due to more efficient running of the experiment on my part in the second round of the experiment. In any case, in round 2, hot-treated animals do show a change in behavior due to nociception, which, along with the results from the light variable, does suggest learned avoidance. I also found that locomotion was significantly affected by iron temperature. So hot subjects spend significantly less time paused or more time running around the chamber than controls. This pause behavior was reported between approximately 6 and 16 minutes after the subject's last encounter with the iron. So this can't be reflex, but instead it suggests a longer lasting change in motivation, possibly to escape or to find perhaps a more suitable shelter than the dark side of the chamber, which was associated with nociception. So thus far, we have evidence that shrimp's behavioral reactions to nociception go beyond simple reflex, and that these reactions match some of the known adaptive functions of pain, like learning and altered motivation. So now we're going to look at the brain to see if there's chemical evidence to support these behavioral changes that we see. If that nociceptive signal is reaching the brain and resulting in learning and altered behavior, there should be measurable chemical changes that allow them to do so. One of these pain-associated chemicals is nitric oxide. Pain research in mammals shows that nitric oxide helps to transfer pain signals from the site of injury to the spinal cord to the brain. And in two studies, one in crabs and one in lobsters, we've seen that nitric oxide increases in the decapod crustacean brain when they're injured as well. In this 2012 study in shore crabs, researchers showed that a formalin injection to a single claw, but not a saline injection, resulted in an increase in nitric oxide staining on the side of the brain ipsilateral to the injured claw. You can see here the control animal versus this is the saline injected animal versus the formalin injected animal. This is 10 minutes after injury and 30 minutes after injury. You can see an increase in the optic density of nitric oxide occurring in the brain here. In another non-pain or nociception related study, Benton, Sandeman, and Belts found that nitric oxide increased in the brains of American lobsters after unilateral antennal ablation, again on the side ipsilateral to injury. So this is the control image here, and 24 hours post-ablation. This is in the olfactory pseudocerebrum of the animal. And you can see here, at least in cell cluster 10, there's a large increase in staining of nitric oxide here, as well as a brighter staining in the accessory lobe of the animal. Nitric oxide increases in mammals, crabs, and lobsters with injury. So I wanted to see if it was the same for shrimp. Nitric oxide is a gas which only lasts a few seconds. So research, including the studies that I just showed you, focus on its synthesis protein, which is called nitric oxide synthase. The one that I used in this study is called universal nitric oxide synthase, which is a mouthful, which we're going to call UNOS. So the prediction for this experiment was that treatment with the hot iron would result in an increase in UNOS in brain regions associated with learning, memory, and sensory integration. Or again, those areas that might be involved in pain processing and responsible for the behavioral outcomes we see, as well as a felt experience that we think of as. It's worth noting here that nitric oxide has many roles in the brain. So we're not looking for presence or absence of this chemical, but for an increase in the hot group as compared to controls. And I think you can see that in this study here. There's nitric oxide kind of all around this brain, but there is an increase that is associated with injury. To measure levels of UNOS in the brain, I performed an immunofluorescence assay with 12 subjects that I took from the same group that I used for behavior. I developed a humane eugenol euthanasia method working from several studies in other shrimp species. If you're not familiar, eugenol is the active chemical in clove oil, which has been shown to be an effective anesthetic in some crustaceans, including in shrimp. The method was essentially an anesthetic dose followed by an anesthetic overdose. And this allowed me to ensure that I wasn't introducing pain in the euthanasia step of the procedure, to maintain a no pain group in order to compare to the pain group. I performed immunofluorescence to stain brain tissue with antibodies for UNOS, our pain-associated biomarker, as well as for synapsin and DAPI, which were both chemicals that allowed me to better visualize the brain structure and locate the UNOS within it. Using fluorescent images of the stained neuroanatomy, I developed a method to quantify UNOS in 4 regions of interest in the brain. And now we're going to look at those brains. Okay, so this is a large figure. I can get my laser pointer to work. Okay, so on the left, here we have micro CT images of a related Alpheus species that I've adapted from a recent paper. They show all of the neuropils of an Alpheid shrimp. Neuropils are these dense masses of projections where cells process information and talk to each other through synapses. In my images of Alpheus angulosus here on the right, these are the yellow-green areas here. They are yellow because they are green UNOS along with the red synapsin. Not shown in the maps on the left are the clusters of cell bodies, which are stained blue with DAPI in my images. The cell bodies originate all of the projections that make up the neuropils. So this is important because I quantified my, the quantification method that I developed was developed specifically to measure UNOS in the cell clusters. So all of my regions of interest, or ROIs, will be these blue cell clusters. My ROIs are from 2 areas of the brain. The top right image here shows a representative section from the protocerebrum, which is located about here in the brain. These large structures are the mushroom bodies, the sensory integrative learning and memory centers of the decapod crustacean brain. These structures will be familiar to insect brain folks as well, and have previously been referred to as the hemiellipsoid bodies in decapod crustaceans. Nicholas Strausfeld, Gabriel Wolf, and colleagues showed in 2020 that these structures are homologous to the mushroom bodies in insects, and they urge researchers to use this nomenclature in future publications. Around the mushroom bodies, I quantified UNOS in 2 ROIs, which I've termed the dorsal and the ventral globuli clusters. These cell clusters have been shown in related species to send their projections into the mushroom bodies. So they are likely to be involved in learning and memory. So for this reason, I predicted that the hot iron would result in an increase in UNOS in these regions. The second area of the brain I was interested in is located about here in the brain, the olfactory deuterocerebrum. And it contains these prominent olfactory lobes, which they process chemical information from the shrimp's antennules. So the first ROI that I quantified in this area, I termed the projection olfactory cluster. Projection because it actually sends its axons, projects its axons to form the olfactory globular tract. This olfactory globular tract sends information from the olfactory deuterocerebrum to the protocerebrum to the mushroom bodies and other higher order centers of the brain. So it's thought to be involved in sensory integration. Additionally, in the crab and the lobster studies that I showed you earlier, they found that nitric oxide increased in this area of the brain in response to injury. So for these reasons, I predicted that the projection olfactory cluster would also show an increase in UNOS in response to the hot iron. The last ROI that I looked at is I termed the local olfactory cluster. Also associated with the olfactory lobe. However, unlike the POC, the LOC, the extent of the LOC's function is local to the deuterocerebrum and the olfactory lobes. It is clearly stained with UNOS here because nitric oxide is also involved in olfaction in decapods. But it is unlikely to be involved with sensory integration, learning, or memory. So I predicted that this area would not show a difference in UNOS between hot and control groups. To quantify UNOS within these ROIs, I used deep learning software to train a classifier to differentiate stained cells from background, and I created a mask for each image. From these masks, I measured percent area stained, which was equal to the masked area, or these white pixels here, divided by the total area of the ROI within the yellow border. This was the dependent variable that I used to compare dreaming groups. And here are the results. So here we have box plots of the hot treatment in red and the control in gray, showing the percent area stained of each ROI. The green checks here again mean that I predicted an increase in UNOS staining due to treatment. And the red X indicates that local olfactory cluster, which I predicted would not differ due to treatment. Each data point here represents an average for each subject for each ROI. So I began by running a linear mixed model to compare treatment groups across all three areas of learning, memory, and integration. And I found that treatment with the hot iron resulted in a significant increase in UNOS staining across these regions. I then followed up with planned post-hoc contrasts for each ROI and found that the ventral globuli cluster shows a significant increase in UNOS with the hot treatment. These cells project into the learning and memory center of the brain, into the mushroom bodies, supporting the behavioral evidence that shrimp can learn from nociception. UNOS in the dorsal globuli cluster did not show a significant difference, but the effect was in the same direction. So it's possible that with an increased sample size, it would show an increase. It's also possible that this area has a different function than the VGC. For the projection olfactory cluster, thought to be involved in integration of sensory information, the treatment effect was borderline significant at p equals 0.06. This increase in UNOS for the hot group is in line with the previous studies in crabs and lobsters, where nitric oxide increased in this area in response to injury. Finally, because I didn't expect treatment to have an effect on the local olfactory cluster, I analyzed it separately with a simple linear model, and I confirmed that there was no treatment effect. Hot and control were barely different from each other. Obvious, I think, from this graph. These results are consistent with my predictions and with the shrimp's behavior in response to nociception. Let's have a little summary. So we began by confirming that the hot iron used in experiments did cause nociception by showing that it altered shrimp reflex behavior and human perceptions of that behavior. We then saw behavioral evidence of nociceptive integration. Shrimp learned from the noxious heat, and they displayed other non-reflexive behavior in response to nociception. And finally, we saw evidence for this integration in the brain through, in part, an increase in nitric oxide, which is localized to higher order or integrative areas of the brain. So overall, these results provide evidence to support the hypothesis that shrimp display nociceptive integration, ruling out the alternative hypothesis of mere reflex and supporting the potential for pain in shrimp. So looking back at the criteria from Jonathan Birch, Andrew Crump, and colleagues, here are the places where I think this study fills in some evidence gaps for Caridian shrimp. So criterion 1, nociception. Though I did not perform physiological recordings, this study provides behavioral evidence to strengthen the argument for nociception in these animals. Criterion 3, nociceptive integration. This was the focus of the study and clearly the place where it provides the most evidence, as I just described. Criterion 5, motivational trade-offs. In choosing to avoid the dark side of the testing apparatus, shrimp were also giving up a critical resource for survival, the protective shelter of the dark, which is something that Bob Elwood discusses at length in his recent paper as a type of motivational trade-off associated with the idea of pain. So, though this study didn't set out to explore motivational trade-offs, I think the results here are suggestive of the capacity of Caridian shrimp to make complex decisions between competing motivations. So future studies might consider adapting the behavioral paradigm here and varying the level of light on the light side of the chamber or the intensity of the nociceptive stimulus to explore possible motivational trade-offs further. Finally, criterion 7, associative learning. In condition-place avoidance trials, shrimp learned to avoid their preferred compartment when it was associated with nociception, providing evidence for associative learning with a cost for Caridian shrimp. But that's really awesome for Caridian shrimp. What does this mean for Penaeid shrimp? Again, these are the shrimp most often farmed and consumed by humans, and they also have the least amount of pain or sentience research. So first, evidence for sentience in Caridian shrimp suggests that the capacity of pain is not limited to reptantia. So several authors have noted that evidence for more species of decapods serves to strengthen evidence for sentience across decapod crustaceans as a group. Along with the very limited research on Penaeid shrimp, one of the most cited arguments against their sentience has to do with proposed constraints on their neuroanatomy. So I just want to quickly address these arguments here. The first component of this argument is the relative size of their mushroom bodies. So this figure from a 2020 paper by Strausfeld and colleagues shows functional brain structures in Malacostraca with mushroom bodies in magenta. So you can see here in Dendrobranchiata, the suborder that contains Penaeid shrimp. The mushroom bodies are relatively reduced in contrast to Caridian shrimp here. Here's Alphidae and Reptantia. And the authors argue that the size and the complexity of the mushroom bodies that they saw in this experiment in Malacostraca is related to place memory and the complexity of the environment that they live in. So Penaeid shrimp live out in schools in a mostly featureless open ocean, thus their mushroom bodies are reduced. However, they still have mushroom bodies. In this paper, the authors identified these structures by staining for a gene product, which in insects is required for learning and memory. So the retained presence in these animals suggests a retained capacity for learning and memory. And there's no evidence yet to suggest that the reduction in these structures precludes nociceptive integration or pain perception. The other part of this brain constraint argument is that Penaeid shrimp are missing the accessory lobes, which are present in the Reptantian species, which have been hypothesized to be involved in pain processing in these animals. However, Caridian shrimp are also missing the accessory lobes. And my work here has shown that accessory lobes are not necessary for nociceptive integration or for pain behavior suggestive of pain. So I think it's possible that an alternate structure perhaps the olfactory lobes serve an analogous function to the accessory lobes in shrimp. So it kind of leads to another question. If Penaeid shrimp are hanging out in the water column, they probably can't learn to associate place cues with nociception. Maybe they can't learn to avoid things or animals or places that hurt them. So why then would they feel pain? What's the adaptive function here? Well, pain may serve other adaptive ecologically relevant functions. So for example here, Robin Crook and colleagues found that when schooling squid were injured, they exhibited heightened sensitivity to predators, allowing them to quickly escape and survive. But when nociception was blocked and they were not able to feel that injury due to a localized anesthesia, their reaction time as well as their survival decreased. So I think it's possible that pain may serve a similar fitness enhancing function in Penaeids, perhaps a heightened sensitivity to predators or something else that we just haven't discovered yet. So I hope that I've shown here that Penaeids certainly aren't ruled out of the pain picture, but we really need more research on these guys, especially due to the extent of their global exploitation. The behavior study that I used in my experiments is unlikely to be effective for Penaeids, again, due to their ecology, but I think that the neurochemical study could be replicated effectively in these animals. With that, I want to thank the IWRS for their generous support of students, along with my other funding sources, the Joanna Foundation and the College of Charleston. My co-authors, Dr. Hughes and Dr. Ruscio, and I are currently working on the publication of this research. But if you want to learn more in the meantime, feel free to reach out to me via email, or you can access my full thesis on my LinkedIn profile. And that's all I have for you today. I'm very interested in thoughts and questions from this super smart, invertebrate-loving audience. Thank you.
Thank you so much, Jesi. That was a fascinating presentation. I learned lots of things. I'll start with a question from the audience. So this says, forgive my extreme ignorance, mammalian researcher, but was the shrimp submerged in water during the CPA testing or nociceptive testing? AKA, did the water heat up as well?
Yeah, that's a great question. Yeah, I didn't mention that. So I actually drained the water out in order to perform the touch bouts. So the experimental treatment was performed in a dry chamber, but their acclimation and the testing was done in a filled chamber. So when I needed to touch them, I drained the water and then replaced it.
Great, thank you. So another question for you. When you were doing your neurobiology study, why focus on the cell clusters and not the neuropil itself?
Mm-hmm. Yeah, this was a really difficult chemical to work with because it's so ubiquitous in the brain. So when you look at you can almost see the entire structure of the brain just with the UNOS fluorescence. So it's kind of everywhere. It was really difficult to devise like how I was going to quantify anything in the brain. But I found that looking at the neuropils was just too difficult because they were so ubiquitously stained. However, when I was looking through, I could look at the cell clusters and I could see that some of them were different from others. Some were brighter and some were lighter. And so I thought that was a good place to go. And doing the quantification, the method that I used really only worked for one or the other. Maybe someone who knew what they were doing with image analysis before this study or like knows what they're doing with image analysis can help me with looking at the neuropils. That would be awesome. But yeah, this was the direction that I decided.
Cool. Is there any evidence in shrimp that the tail flips or the autotomy that you saw require integration, central integration, and aren't just lower ganglion reflexes?
So I measured them as reflex reactions. So when I was measuring those particular behaviors, I was just looking for evidence of those lower um, ganglionic reactions. Okay, so I was really just looking to see if the hot iron caused the reflex, just to make sure that like the stimulus was actually effective. It was hot enough, and actually nociceptive to the species. So I really was only looking for that lower order um, reaction. I don't think that we do have evidence that it requires central integration. There's some like observations from one of Bob Elwood's papers. They discuss in detail behavioral evidence that suggests that claw drops occur as a reaction, but also perhaps as a pain associated behavior because they occur after some time.
Yeah, it would be interesting to investigate that to determine whether their reflexes will require some central integration, just because we have some studies in cockroaches, Emmanuel and Libersot, like 2019, I think is the paper, where they show that if you do like a cut of the neck connective, so you can't actually send signals anymore between the brain and the body, but otherwise the animal is intact, you can still get startle responses to nociceptive stimuli in the body, but you can't get a full escape behavior. So they startle, but they don't actually escape. And to me, that's a really interesting example of like, okay, what actually requires the brain versus what can just be performed by the abdominal ganglia of the cockroach, or the ventral nerve cord of the cockroach, I suppose. So it'd be interesting to know if either of those behaviors require any kind of signaling from the brain proper, the protocerebrum and deuterocerebrum of your shrimp or not.
That would be interesting. That seems very relevant.
Another question. Round two.
I'm so sorry, Megan. You
Oh,
cut out for me there. Could you ask that again?
I know my internet, it's terrible. You'd think in a faculty office, how long was the interval between and testing in round one? And did you train the shrimp again before round two?
So I did not do any additional training of the shrimp. Let me go again. I think I have my, just to answer this question better. So the trials were the training. So conditioning occurred during the trials, which occurred in threes. So the animals were again put into acclimation. I gave them that experimental treatment and then they chose. So this was all a 16-minute process. And then the animals were put back and done again for 16 minutes. All the measurements were taken during these choice tests. And I'm sorry, cut out a little bit again. What was the question about the time between?
So how long was the interval between experience and testing in round one? And did you train the shrimps again before round two?
Oh, okay. So training and testing, about 6 minutes between, I guess, performing the experimental treatment and allowing them the choice. But I suppose they were being trained through all three trials. So it's a really complex experiment. One of the things I would do differently is making this simpler if I could do it again. So
Another
yeah, and
question
then
for you.
same. Oh, sorry. Follow up to that, just to fully answer the question. Same thing happened in round one as round two.
So it looks like you have three rounds of this experiment, but you presented data from round one and round two. What happened to round three?
So again, very complex to explain. Round one includes three trials. Round two includes again three trials.
Got it.
So yeah, trials, rounds. Yes.
Yes, on it. Um, this question is about the pinia shrimp. So the ones that you talked about at the end.
Mm,
So you were talking about how they, and this is from me, you were talking about how they have, this open, featureless ocean. And so maybe they don't need as much place learning.
mm.
I wondered if there could still be other ways that they could think of place or the space around them. So, like one thing I was thinking, if they're in these big schools, is their place actually relative to where they are in the school? Because it's not featureless if there are thousands and thousands of shrimp around you. Potentially, there's something to do with the density of your environment in relation to the other members of your school. Olfactory cues, although I think I recall that pinia shrimp are not big on olfactory cues. Um, I just wonder if there are still ways that these animals could conceive of their place in space, just in a different way that we, as very visually oriented, feature full environment animals, tend to think of space.
Yeah, no, I agree. I certainly wouldn't argue with that point. I think I was kind of going off of the author's argument in the paper. That was their suggestion, kind of the pattern that they saw. Benthic animals having a larger mushroom body, social animals having larger mushroom bodies. But again, they retain that mushroom body, so they're using learning and memory for something. And I mean, I'm sure that what you're suggesting is completely plausible. I think if somebody could create an experiment to test them on that, that would be awesome.
Another kind of follow-up related question to that. Are these particularly mobile shrimp? Like, do they swim around? They still have legs? The pinia shrimp?
Yes, they have legs. Yes, they can swim around. I think it's just that they don't necessarily, like the snapping shrimp have burrows. They have that they defend as their territory. They have monogamous mates.
Oh, cute.
And so, they've also been shown to have individual recognition. So they're like, okay, here's my house, there's Bob's house, there's Jeff's house. So I think, them having these really complex learning and memory centers, these huge learning and memory centers that take up like 3/4 of their brain,
For
makes
sure.
sense for them to hold all those uh, memories in because they have conspecific aggression and things. There's a lot of reasons for them to have those large structures. Um, I don't know as much about Canadian shrimp because I haven't studied them. I just kind of wanted to refute some arguments that I've seen um, based on what I've learned.
Yeah, love it. Well, that is all the questions that we have and also all the time that we have. So that's convenient. Thank you so much for your time and your attention. Thank you, everyone who came today to learn about this work. Again, you can find all of Jesi's work as a very convenient PDF on her LinkedIn page. So please feel free to visit that. And I hope that you'll all join us for our next seminar coming up soon. Thank you so much. And thank you, Jesi.
Thank you all.
Bye.